Good morning, all. Today, in this class, we are going to study about the cerebrospinal fluid. It is one of the very important fluid and it is a part of extracellular fluid. Cerebrospinal fluid, or in short, we call it as CSF, is the clear, colorless, and transparent fluid that circulates through the ventricles of brain, subarachnoid space, and central canal of spinal cord. It is a extracellular fluid. So these ventricles of brain, you can see here, ventricles of brain are nothing but these are the cavities here inside the brain, and these cavities are interconnected. They are connected to each other, and these cavities, they contain cerebrospinal fluid, because these cavities, that is ventricles of brain, they have got a specialized type of cells called as, this is a plexus of cells. Many cells are there. There is a cluster of cells. So these cells together, they are called as choroid plexus. So this choroid plexus, which is there in this ventricles of brain, this choroid plexus, it secretes cerebrospinal fluid. So ventricles of brain is nothing but these are the cavities which are connected to each other. There are totally 4 ventricles of brain are there. There are 2 lateral ventricles, as you can see here in the diagram, and there is one thing called as third ventricle and fourth ventricle. So totally 4 ventricles are there in the brain. Here you can see in this diagram lateral ventricles. This is the anterior view and this is the lateral view. There are 2 lateral ventricles. There is a third ventricle, here you can see, and there is a fourth ventricle. So there are totally 4 ventricles are there in the brain, which are interconnected cavities, and they contain choroid plexus, means these are the cells which secrete CSF. So these ventricles, they contain cerebrospinal fluid. So here you can see it appears like this. These are ventricles. They will be appearing like this. So this diagram is showing all the 4 ventricles. The next thing is about the properties and composition of cerebrospinal fluid. First one is about the properties. Volume 150 ml, that is, it is between 100 ml to per 200 ml of cerebrospinal fluid will be present in these ventricles, spinal canal. Rate of formation, it is formed at a rate of 0.3 ml per minute. The specific gravity of this CSF is 1.005 and reaction of this CSF is alkaline. The next thing is about the composition of cerebrospinal fluid. Cerebrospinal fluid, 99.13% of it contains water and remaining 0.87% contains solids. These solids are again divided into organic substances and inorganic substances. The organic substances here are like proteins, amino acids, sugar, cholesterol, urea, uric acid, creatinine, and lactic acid. And the inorganic constituents here are like sodium, calcium, potassium, magnesium, chlorides, phosphate, bicarbonate, and sulfate. These are the inorganic constituents. The next thing is about the formation of cerebrospinal fluid. Cerebrospinal fluid is formed by choroid plexuses situated within the ventricles. Choroid plexuses are tuft of capillary projections present inside the ventricles and are covered by pia-meter and ependymal coverings. A large amount of CSF is formed in the lateral ventricles. The next thing is about the functions of cerebrospinal fluid. The first function is a protective function. Cerebrospinal fluid acts as fluid buffer and protects the brain from shock. Since the specific gravity of brain and CSF is more or less same, brain floats inside the CSF. When head receives a blow, CSF acts like a cushion and prevents the movement of brain against the skull bone and thereby prevents the damage of brain. So CSF has got a protective function. It acts like a cushion to the brain. Then the second function is regulation of cranial content volume. Regulation of cranial content volume is essential because brain may be affected if the volume of cranial content increases. It happens in cerebral hemorrhage and brain tumors. Increase in cranial content volume is prevented by greater absorption of CSF to give space for the increasing cranial contents. So here what happens, if there is cerebral hemorrhage, there will be collection of blood in the brain. If there is any tumor, there will be a space will be occupied inside the brain. So skull cannot enlarge in size because it is bony in structure. Now when skull cannot enlarge in size, then how that space will be occupied? If the amount of blood increases there, if amount of blood is there or some amount of hematoma is there or a tumor is there, they will occupy some space here in the below the skull in the brain. So if they have to occupy some space, something has to be compromised here. So in such condition, what happens? The CSF is reabsorbed here by the ventricles. So when CSF is reabsorbed, so say for example 100 ml of CSF is reabsorbed, so that tumor or that hematoma or that blood will get some space there inside the skull. Otherwise what happens, if it does not get this space, then it will increase the intracranial pressure. There will be lots of pressure over the brain if it is not reabsorbed, and that will lead into symptoms like epilepsy, convulsion, unconsciousness. All these symptoms will come. So this is one of the important function of CSF, that is regulation of cranial content volume. So whenever there is a need, if the space there inside the skull is increasing, so this CSF is reabsorbed to provide space for this space occupying lesions inside the brain. Then the third function is medium of exchange. CSF is a medium through which many substances, particularly nutritive substances and waste materials, are exchanged between blood and brain tissues. So it is a medium of exchange through which nutritive substances and also the waste substances are exchanged between the brain and blood. The next one is about blood-brain barrier, that is BBB. Blood-brain barrier is a neuroprotective structure that prevents the entry of many substances and pathogens into the brain tissues from blood. So anything from blood cannot enter directly into the brain. These substances are protected from entering into the brain. There are many substances are there. Many substances, many organisms, many ions, they are protected from entering directly into the brain. So it is believed that there is a layer between brain and other tissues which protects them. That layer itself is called as blood-brain barrier. It was observed more than 50 years ago that when tryptophan blue, the acetic dye, was injected into living animals, all the tissues of the body were stained by it, except the brain and spinal cord. When the tryptophan blue is injected, all the tissues were stained except brain and spinal cord. This observation suggested that there was hypothetical barrier which prevented the diffusion of tryptophan blue into the brain tissues from the capillaries. This barrier was named as blood-brain barrier. It exists in the capillary membrane of all the parts of the brain except in some areas of hypothalamus. So because of this experiment, now it is believed that there is some protective layer is there between the blood and the brain tissue and spinal cord, which prevents the entry of many ions or many substances into the brain, which is called as blood-brain barrier. So this is about the cerebrospinal fluid.